All right, so today we're going to be doing the endospore stain. So you can see that I actually have a beaker of water sitting on a hot plate with a stirrer inside, as well as some wire mesh up here. So the whole point of the endospore stain is we want to stain the bacterial cells to see if they have endospores. So it's actually going to require the slide sitting on top of basically steaming water. So it's not boiling, but you can see it's producing steam, and that steam is going to be hitting that slide. So the first thing I went ahead and did, which you can actually see, is I did a bacterial smear. So that bacterial smear, you can see it right there, kind of toward the edge of my finger. That is our bacteria that we are going to be staining. So what I'm going to do is I'm going to place the edge of that slide so that way it's a little easier for me to grab. That slide is going to get a little hot. So once I do that, I want to add malachite green. So malachite green here is a green stain, and what's going to do is it's going to stain those endospores. And so I'm going to add a couple of drops to this, and I'm going to let that sit on top of that beaker for 5 minutes. So as that beaker is producing that steam, and because it's warm, it's going to actually end up causing evaporation. So I want to stay close to this and continue adding some of that green set, that malachite green, to that slide for 5 minutes. So we're going to let that sit for 5 minutes, and you'll probably see me add a couple of drops of malachite green throughout that, and then we're going to continue with the staining process. All right, so what the heat has essentially done is it's softened up those endospores a little bit to take up the stain, but also it has gone ahead and stained the cells themselves. So now this slide is actually relatively hot. You can see the evaporation there that has occurred, and so we're going to move that out of the way just slightly so that way we can finish the rest of the stain. So what I'm going to do is, because this stain is very, very similar to the gram stain and we have another step or two, I'm going to grab the slide with the clothespin. And so what we need to do now is we need to rinse the slide. So I've basically got my distilled water here. Same thing that we did with the gram stain, is we're just going to rinse the slide. And actually, as I'm doing this, you can see that green running off. But if I pull up the slide, get rid of that background there, there's a little bit of green right there. So that little bit of green is telling me that the stain actually took. So that's great. So the next thing that we're going to do is we're going to counter stain. So what we hope has happened is that the endospores have retained that green coloration. So now the cells are probably going to be clear, which means we are going to have to counter stain. So we've seen this one before, safranin. We use it in the gram stain. It's the last step in the gram stain. We are going to take a drop of that, drop or two of that, put it on the slide, and we're going to let it sit for 30 seconds. So after that 30 seconds, once again, we are going to rinse that slide off. And now we've basically completed that endospore stain. So we've used the two stains, the malachite green for 5 minutes under that steam, and then we used the safranin. So the last thing we have to do is we have to blot dry. And remember, whenever we blot dry, we want to make sure that it is completely dry. We don't want to smear, but we want to blot. So I usually take the blotting paper, fold it in half, put my slide in there, and then just gently pat. And the whole point of this is to make sure that we get rid of the water on the slide, because remember, we are then going to use oil immersion. And so if I put that back on there, you can see where that stain is, right here in the center. So we see a little bit of green and a little bit of pink. And so what we're looking for is, again, those endospores. And so we want to make sure that we look at that underneath the microscope, because some of those little parts might be just clumps of stain. So once again, looking for those endospores. Those endospores will appear green, and if it's a relatively young culture, meaning it's been in the incubator and the cells are like 24 hours old, those endospores will actually be inside of the cells. If the culture is more than 24 hours old, what will end up happening is those endospores will end up being ejected, so we'll see the endospores around the cells. So again, now that it's dry, we're going to stick it on the microscope and we're going to look at it under the oil immersion lens.